Seeing as I've had you do case studies, I thought I'd run through a few of them. What you need to know is obviously disease name, causative pathogen, the life cycle, the method of transmission, the effects on the host, which are the signs and symptoms, methods of treatment, and methods of control. Control is prevention mechanisms to stop people from getting it in the first place. In between signs and symptoms and treatment, we like to also do the host response. So if I start off with looking at a disease like rubella, the disease name is rubella. Now, interestingly, Ella comes from the Latin, which means little, and rub means red, like ruby, for example. So rubella's other common name would be German measles. Doing your summaries for your disease, you only need to have the brief outline of it. So working with that, you just flow down through these 8 points and it's reasonably straightforward. The causative pathogen for rubella is the rubella virus. The life cycle. Now remembering when you're talking about life cycle, you're dealing with whether it is direct or indirect in its method of transmission. That is to say, does it have an intermediate host that is different in its species from the final host? So the life cycle for rubella is direct because it goes from person to person. The method of transmission is through, now this is where it gets confusing because we again use the words direct and indirect as a consequence of looking at transmission. So for this, the transmission is through direct contact. This is choosing from things like air, water, vectors such as mosquitoes, fomites for your indirect contact, which you can also get. So when we say direct contact, it might be literally touching someone when you've got the pathogen on your hands. It might also be along the lines of coughing and sneezing and airborne droplets that are directly inhaled by someone else. The indirect contact is using the abiotic vectors, so fomites in other words. Signs and symptoms for the disease. This one is a typical one for all viruses, which means that it's a sore throat, headache, runny nose, fever. They are typical for most every virus. Methods of treatment. Usually with treatment, what you're doing is you're looking at what are the things that we do to combat the signs and symptoms of the disease. So for viruses, you use liberal fluids, that's to try and offset some of the symptoms, particularly the headache, paracetamol or other analgesics. Analgesics just means painkiller. And rest. We often tell people, go home, stay home until you feel better, because your body actually needs the opportunity to try and rest, to put all of its resources into just fending off the pathogen that's affecting your cells. The method of control for this disease is vaccination. Now, going back to the signs and symptoms, these are the ones that are typical for all viral pathogens. But in the case of rubella, you also have a rash of red spots that starts generally around the throat and the neck, goes onto the face and protrudes down the arms. Can also go on all the extremities. But the red, the rash is made up of red-pink spots, and that's where it gets its name rubella from. So your methods of control, the vaccination is your primary mechanism. In Australia, we have a whole range of different vaccination programs which begin as an infant. Aside from vaccination, you will also look at quarantine for individuals that are infected to stop the spread of the disease. It's a really high virulent disease. It's got an R naught somewhere in about 10, which means that every one person that's affected would affect 10 people if that was the R naught of this particular pathogen. I don't know that it's 10, but it's high like that. It's an extremely contagious disease. So remember when we looked at the word infectious, so under the umbrella of infectious diseases caused by microscopic pathogens, something could be contagious, or it might just be communicable. So it depends upon how, what degree of virulence it has. So the host response is what does your body do to try and combat it? The increase in temperature, and as well as increase in temperature, you will produce antibodies to the pathogen. But the production of antibodies is slow, and it usually occurs after you've already had the signs and symptoms of the disease. So it lags behind in terms of the way that develops. So if we drew a graph of the introduction of the pathogen, so if this is the antigen coming into your body, antigen starts to build up its numbers. As it builds up its numbers, you become symptomatic and you start to display all the signs and symptoms of the disease. But at the same time now, if we looked at what's going on with your antibodies, you had no antibodies and this curve lags behind, and you start to build up your antibodies, but the symptoms are already there. The antibodies start to kick in, and as a consequence of the antibodies getting higher, the amounts of antibodies in your blood the amounts of antigen in your blood gets lower. So that's as a consequence of these antibodies that will then stay in your bloodstream, giving you, hopefully, lifelong immunity against the pathogen. And that would be the summary for German measles.